Hello everybody. In this video I want to explain more about the research we did in grapes, our research in our database and our research at a trial location. And topics in this presentation are the relationship of nutrients with the sugars in plant sap analysis, the relationship of nutrients with the pH in plant sap, and deficiency symptoms and the corresponding values in plant sap. And I want to share the results of this research in this video. First, a small introduction to NovaCropControl. We are a lab where we test plant sap analysis, but also water analysis on nutrition. And with every sample, you get 21 parameters tested. We have target values for grapes, and we also have our own research location of 1,000 square meter of plastic tunnel for independent research, and 3 indoor climate chambers where we also do the whole year-round research in finding the relation between the health of the plant and our plant sap analysis. We are located in Oostenrijk, that's in the Netherlands. And we're active in 55 or more than 55 different countries. So shipping from all countries are possible within 4 to 5 days. And once a package with samples arrive in our lab, results will be sent within 24 hours after arriving. Consultancy on the analysis results are also possible. And in the form of help with the interpretation of the data. And we also give courses like seminars or webinars where we explain more on the nutrient uptake by a plant. So which nutrients do we test? So with every plant sap test, we test 21 different parameters. And it's good for later in the presentation to know that if we talk about nitrogen, we measure with plant sap the nitrate separately, the ammonium separately, and we measure total nitrogen. And total nitrogen are basically all different nitrogen forms. So in total nitrogen, there is a nitrate, there's ammonium, there are amino acids and proteins. And that all together is what we call total nitrogen. Proper sampling is essential. So there are some key points to be aware of on the moment you start taking leaf samples for grapes. One of them is sample early in the morning, so best before 9 a.m. because then the leaves are not affected yet by the heat or the light intensity of the sun. Sample young and old leaves separately because that will tell more about the mobility of the different nutrients, and it will tell more about the transport of the nutrients from the older parts to the younger parts. We have a manual on the website for grapes on how to sample. Sample leaves without the petioles. Petioles are for transport, so we only want the leaf blade because that's also where the photosynthesis takes place. Avoid side parts or the head or of the field because there's always more influence by sun, wind, etc. Sample deficiency leaves separately from healthy leaves and free from dew, moisture, or dirt. Or if you have done any foliar application, sample before or at least 1 week after. And the main reason for that is that there always will be residue on the leaves. And on the moment we press the sap out of the leaves, also everything what is on top of the leaf will get within the sap, and that can influence the results. Always pick for the young leaf sample the youngest fully developed leaf. For grape, this will be the 4th or 5th leaf from the top of the vine, and also pick separately the oldest but still vital leaf opposite to the fruit at the lower part of the vine. Begin with sampling when the shoots are like 40 centimeters, and from then every 2 or 3 weeks until harvest. You can also choose to sample different stages. So for example, when shoots are 40 centimeter with early flowering, early fruit set, with fruit fill, before harvest and after harvest. That are the typical moments when samples are taken. Please use our labels because we can write down all the information on the label. So like the location, the cultivation, the variety, and definitely in the case of the variety, please mention that on the labels, because this helps us with further research on variety differences. And on the label, you can subscribe if it are the young or the old leaves, and you can put some remarks on the labels. Always use Ziploc bags because you can press the air out of the bag, and that's better for containing or the quality of the leaves. Now, the target of the investigation is to get more knowledge in the relation between plant sap analysis and the health of the grape plant, more insight in our grape database built up over the last years, and understand better the antagonistic interactions between the nutrients, the influence of nutrients on the plant sap pH, and more insight in the measured sugars in our plant sap analysis. And eventually we hope that this research and all the knowledge coming from it helping our clients getting more out of their plant sap analysis. Now, the database research is based on almost 6,000 grape analysis from the last 2 years. And basically what we did is we taken from every element a range of ppms where a certain number of samples are found. So like in this example, a range of 200 ppm. So and within this range of potassium, I took an average of the pH values measured, what we see over here. And in the study, measurements were only included if at least 100 different analyses fell within a certain range. So like we see over here, not enough samples are collected where potassium is in a range of 700 to 900 ppm. So I didn't took these further in the investigation. And like we see over here, like the higher the potassium goes, the higher the pH values measured, so the average pH measurement. And so if we put that in a graph, we see that as well, that the higher potassium goes, the higher the pH. So this is an example, and I will discuss more on that later in the presentation on something coming out of our database research we did. We also put on that graph the coefficient of determination, which is a number between 0 and 1 that indicates the extent to which a statistical model is able to predict a particular outcome. So the closer it gets to 1, the bigger the chance it is, like in this case, that if your potassium goes up, that your pH goes up as well. So that's basically how we did our database research. Does that mean that you will always see in plant sap that when potassium is higher, the pH is also higher? No, that will not always be the case. In theory, the chance of that is high, which is also indicated by the data, but in the end, it's still an average number of all the measurements in a certain range. So a scatter plot shows what I mean. So you can see here too that a trend is visible. But you can also see that a higher measurement of potassium can also indicate a lower measurement of pH. So it's still a game of elements in the plant, and other elements can have also have influence on the pH, but more later about this. So there are always analyses that cause a deviation due to the damage of the leaves during transport or by leaf foliar spraying, etc. So that can have a major influence on a scatter graph. So that's the main reason why we took an average out of the data in a certain range, because that would show better a trend, what you could expect if you look to the plant sap analysis. Now we also had a trial on location, and we did that at Wine Estate Sint Martinus, which is in the Netherlands. We tested 2 different locations. And on those two different locations, we tested two different varieties, which were the Souvenir Gris and the Cabernet Cortes. Every 2 weeks, we took the young and the old leaves. We also started with a soil balance analysis at the start of the season, and we tested the young fruit and the fruits at harvest. This is an example of the Cabernet Cortes, which we have tested, mostly used for red wines. And this is an example of the Souvenir Gris, mostly used for white wines. So yeah, let's start with the database research. We start with the sugar levels we measure in plant sap. And first of all, I will explain a little bit about how we build up the graphs, which you will see in this presentation. So basically, it's a normal graph where I compared the calcium values, like in this case, with the sugar values. But I also brought in the target values we have for the sugars, which is these two gray lines. So this is the minimum target value, this is the maximum target values, and also put in the minimum and the maximum target value we have for calcium, like in this case. So if we start and we look to the calcium influence on the sugars, then in the young leaves, calcium often has to build up. The main reason of that, it's an immobile nutrient, so the leaves really need to get older or stay over a longer period active to get enough calcium uptake. But we see also that if your calcium stays too low in the young leaves, that your sugars stay behind. So you really need some build up in the young leaves of calcium to get good sugars within the leaves. Also in the old leaves, we see that if calcium is getting too low, if calcium is on a lower value, that the sugars are most likely to be lower as well. But once you get calcium good within the target values, then we hardly see a difference on sugars. So there seems to be a correlation, especially on the young leaves, between calcium and sugars. If we then look to magnesium, yeah, that really seems to be the stimulant of sugars. So the higher your magnesium goes in the leaves, the higher the sugar levels. And that's really showing a very clear trend up. So if your magnesium is too low, yeah, your sugars also stay behind. And in some way it makes sense because magnesium is the central component of chlorophyll, and which is the pigment molecule responsible for absorbing sunlight during photosynthesis. So in plant sap, a very important factor to look at to get higher sugars in the leaves. And especially the young leaves show very clearly that if your magnesium is too low, then there are also almost no sugars produced. So the higher you can get your magnesium, the higher your sugar levels will be in the leaves. And those sugars will be transported to your fruits, and those sugars will be transported to your roots as exudates or to other parts in the plants. So your sugars are very important to look at, and magnesium is the stimulation of those sugars, or help a better buildup of sugars in the leaves because of photosynthesis, or it helps the photosynthesis. Yeah, then the influence of potassium on the sugars. Yeah, it seems to be that the sugars drop when potassium gets too high. And especially in the young leaves, that trend is very clearly visible. And on the old leaves, we see less of a trend. But yeah, young and old leaves combined, we really see that the higher your potassium goes, the lower the sugar levels. And in some way it makes sense because we also see that the higher your potassium is within the leaves, the lower the magnesium levels. And we saw before, the higher the magnesium, the better the sugar levels. So the reason of that is potassium is antagonistic to magnesium. So the higher your potassium goes, the more it will block other cations like calcium and magnesium, like we see over here. So once your potassium is getting too high, that will block your magnesium uptake, and that will have directly influence on the sugar production. So it's not because of potassium itself that there's less sugar production, but it's because the potassium is blocking the magnesium, and that leads to less sugar production. Yeah, then total nitrogen and the relation with sugars. Yeah, most crops we see a link between total sugars and total nitrogen. And also here we see a slightly trend down, but the effect seems to be limited. But if we then comparing magnesium as well with the total nitrogen, then we see a trend up of magnesium with more total nitrogen. So the effect of total nitrogen is not really good visual, and that's basically because with the buildup of magnesium, that's keeping the sugars on a higher level. But we know from other crops as well that we see clearly a trend that if your total nitrogen is getting too high, that it will lead to lower sugar levels. Because basically what does total nitrogen indicate? The higher the total nitrogen, the more water also gets within the leaves. And what does water with sugars? It dilutes it. So the more vegetative your growth is, the more water in the leaves and the lower the sugar levels. But here in the database, it wasn't really clearly visible. But if we then look to the ammonium levels and how those are influencing the sugar levels, then we see clearly that low ammonium gives higher sugars. So if we almost don't measure any ammonium, then we see clearly higher levels of sugars. And especially if we look to the old leaves, if the higher the ammonium goes, the lower the sugar levels. And it makes sense because ammonium eventually is a result of unconverted ammonium into proteins. So it's actually a surplus in the plant. And in many studies, too many ammonium would have a negative effect on the health of the plant. So basically, you want to see your ammonium in your plant sap analysis as low as possible. And you see over here already that it also has a positive effect on your sugars once you can get your ammonium lower in the leaves. Are these results then not influenced by the magnesium like we saw with total nitrogen? No, in this case, the magnesium, so the orange line, doesn't influence the database if we look to ammonium versus sugars. But we know that the higher the total nitrogen, the higher the ammonium levels. So very clear visual that the higher the total nitrogen, the more ammonium we will find back in the plant sap analysis. So basically, that will confirm that if your total nitrogen is higher in the leaves, then it will also lead to lower sugars, because eventually, it comes in a combination with those higher ammonium, and we saw on ammonium a trend down on sugars. So this confirms that also total nitrogen too high in the leaves lead to lower sugars. We also tested that on a trial location. So we took from two different plants next to each other from the same variety, plant sap analysis, and both plants differed constantly on growth. So there was a big difference in growth between the two plants next to each other, and it's also clearly visible on this picture. And if we then look to the long thick shoots, so this one, then we also saw lower sugars compared to the shorter thin shoots and small leaves. And of course, as expected, the bigger the leaves, the more vegetative growth, the higher the ammonium and the higher the total nitrogen, and that was keeping the sugars low. So that also confirms what we were seeing in the database, that higher ammonium or total nitrogen leads to lower sugars. Also iron is important for photosynthesis. So if there's an iron deficiency, the plant will the plant will also produce less sugars. That's clearly visible when we look to the young leaves. The young leaves are also the part where you mostly start seeing the first symptoms of iron deficiency. So the lower the levels of iron, the lower the levels of sugars are within the leaves. And on the old leaves, where normally the iron levels are a bit higher, you don't see any effect anymore of the iron on the sugars. So especially the young leaves are important to look at. So if your iron is too low, or if you see clearly iron deficiency on your crop, you know it's going to be less photosynthesis, and because of that also less sugar production. So those were the trends we saw in our database. And if we compare that with the sugars measured on a trial location, then basically there we saw the same trends. But first, what was quite interesting is that if we look to the Cabernet Cortis, then on location 1 and on location 2, that on location 2, the sugar production or the sugar levels in the leaves were much higher, especially the first half of the season, than on location 1. Interestingly, when we also took samples of the fruits and samples of the leaves on the 6th of August, we saw that the Cabernet Cortis on location 2 showed already the first coloration of fruits, which is interesting because that was also the location where we saw the highest sugar levels. So this confirms as well that if you get a better sugar production in the leaves, that will also go to your fruits, and that can help with, or like in this case, it seems to help also a faster coloration of your fruits, because all those sugars go into the fruits. And if we then look to the magnesium uptake over the whole season, then we see also that Cabernet Cortis on location 2 showed a better magnesium uptake compared to location 1. Yeah, and if we then look to the sugars, yeah, then that confirms that the better the magnesium uptake in the leaves, yeah, the higher the sugar levels. So that what we saw in the database is also what we saw on the trial location. Yeah, and if we look then to the other variety, Sauvignon Gris, then also there we saw the trend that in this case, location 1, the orange line, is showing with the old leaves, which is the straight line, and the dotted leaves are the dotted points are the young leaves. That the Sauvignon Gris on location 1 showed higher sugars, and that was also the first location where we saw coloration. So picture taken on the same moment, same day, and yeah, location 1 showed more development on the fruits than on location 2, and also that could be correlated to the higher sugars we measured in the leaves. So very interesting. But yeah, if we then look to those higher sugar levels in the leaves of location 1, it doesn't match with magnesium, because you would expect that that location would also have the highest magnesium levels. But that is not the case, because the Sauvignon Gris on location 2 showed a much better buildup of magnesium than it did on location 1. So yeah, what has influenced the sugars then? And yeah, we know already that, or what I explained before, it's not only magnesium which can influence the sugar levels, but it can also be nitrogen. Interestingly, yeah, the Sauvignon Gris on location 2 had the highest magnesium compared to all the measurements we did on this other variety and location, but was showing the lowest sugar levels overall. So the only nutrient that could affect the sugars other than magnesium is nitrogen. And if we then look to the nitrogen levels on location 2, here then it clearly shows a big difference of location 2 with the other measurements, that it was much higher on total nitrogen, and yeah, that was lowering the sugars, so that was diluting the sugars in the leaves. So very clearly visible, a big difference in total nitrogen, and that was keeping the sugars low in the leaves, because here again the red line is location 2. So yeah, what we saw in the database, higher magnesium leads to higher sugars, but too high total nitrogen will lead to lower sugars, and that was also clearly visible in on our trial location. Yeah, and as well, if we then look to the total nitrogen and ammonium, yeah, the high ammonium on location 2 was in correlation with the high total nitrogen on location 2. So that makes sense. Also on growth, we saw a difference. So both variety, or the variety Souvenir Gris on location 1 and location 2. So we saw a big difference in total nitrogen, and we also saw that on the plants as well. So location 2 had longer shoots. The internodium between the leaves were much longer than what we saw on location 1, where the leaves were more closely to each other and the shoots were less stretched out. So yeah, that difference in total nitrogen was also clearly visible on the crop itself. So yeah, conclusion on this: the higher your magnesium, the better the sugar levels. But if you get or go too high with your total nitrogen in the leaves, that will lead to lower sugar levels. But magnesium is really the stimulation of higher sugars in the leaves. Now, we also did a database research on pH we measure in plant sap. And yeah, over here, in the beginning of the presentation, you saw it already. The higher the potassium goes, the higher the pH in plant sap. So very clearly visible that effect is there. So the higher the potassium, the higher the pH in plant sap. And that's also something what you will then see in your fruit. So if you get higher pH in the leaves, most probably you're also going to get higher pH in the fruits. And your potassium levels will be higher as well in the fruits. Yeah, if we now look to the calcium versus pH, then it's interesting to see that especially when your calcium uptake is too low, that leads to a higher pH in plant sap. Yeah, and that can be correlated to the higher potassium in the leaves, because we see very clearly that if your calcium is lower in the leaves, that gives more room for potassium uptake. Or if your potassium is very high, it blocks the calcium. So that's that antagonistic interaction. So the lower the calcium, the higher the potassium. So you really see that potassium goes way higher on the moment your calcium is getting too low. And because of that, that leads to a higher pH. So, and the other thing is what we saw: if your calcium is getting too high, or too high, it's building up with high levels in the plant, it can lead to a slight buildup of pH in the leaves. But the effect is not as big as potassium does. Magnesium basically showing the same trend as calcium. Here as well, again, when your magnesium uptake is too low, that will lead to higher potassium in the leaves, and that's the reason why it's pushing the pH levels up as well. So also with magnesium, too low magnesium leads to more potassium, and the higher potassium leads to a higher pH. Ammonium and the effect on pH. Ammonium just seems to have a small influence on pH, so there's a slight trend up, but it's not clearly visible in the data. Eventually, you always want to have ammonium in the plant as low as possible, or best even around 0. Yeah, and if we look then to total nitrogen, yeah, then the total nitrogen provides also a higher pH in the leaves. But also potassium enters the plant more easily, because if we look to this graph, we also see that the higher total nitrogen goes, also the potassium levels seem to build up in the leaves. So the question is over here, is the total nitrogen leading to a higher pH, or is it the potassium which leads to a higher pH because it's better in uptake with higher total nitrogen? That's not clearly or not really clear, but I expect that it might be the potassium leading to a higher pH, and that it's less the total nitrogen. But also with total nitrogen, you see a trend up of the pH levels in the leaves. Yeah. Interesting, then if potassium has so much influence on, for example, your magnesium uptake, your sugars, and on your calcium and pH, if we put that all in one graph, then we really see that if your potassium builds up in the leaves, it will lead to lower sugars because it leads to lower magnesium, and it leads to a higher pH. So potassium seems to be very important if we look to plant sap analysis to keep an eye on, because if your potassium is getting too high in the leaves, it has directly influence on the magnesium, it has influence on the sugar levels, and it has influence on the pH. So potassium is something to keep an eye on in plant sap analysis. Now, and if we look then to our database and we compare the potassium with the pH, yeah, then potassium is also showing a build up around August, and also that's the moment when the pH starts to increase in plant sap. So pH shows the other thing is pH shows a drop in September, and that's also the moment potassium shows a drop in the leaves. So yeah, this really corresponds with the potassium corresponds with the pH levels in the leaves. And what does total nitrogen does? Now, basically, total nitrogen goes fast up around August as well, same we saw with potassium. And the last two measurements, potassium and total nitrogen, comes down again, and so does the pH. So you really see that what we saw in the database, that if your potassium builds up, your pH builds up, and if your total nitrogen builds up in the leaves, that will also influence the pH in the leaves. On the other location, there we saw basically also the same trend. So potassium was overall high in the Souvenir Gris, and the pH also high following the same trend. Potassium in the first 2 months much lower in uptake on location 2, which leads to a lower pH, but later when potassium builds up, the pH increases as well. We might have expected that the Souvenir Gris on location 1 would have had a higher pH, because the potassium uptake is much higher over there. So you would expect a higher pH as well on those leaves compared to location 2. But if we then look to total nitrogen, then we have our answer why the pH is not directly higher on location 1, and that's because the total nitrogen over there is much lower. And we saw that the higher the total nitrogen, the higher the pH in the leaves. And it seems to keep a little bit, or prevents basically the total nitrogen being low on location 1, prevents that the pH is building up too high on location 1. So again, the same trends are clearly visible on a trial location, and if we compare that with our database research. Some other database antagonistic interactions which are interesting to look at, and one of them is the influence of phosphorus on the iron uptake in the leaves, because in various studies we have seen that phosphorus inhibits the uptake of iron. Of course, the pH of the soil also has an effect, so the higher the pH, the lower the uptake. But if your phosphorus is getting too high in the leaves, then it will lead to a lower iron uptake. So always be aware that if you have too much phosphorus in your leaves, then there's a big chance your iron will stay lower as well. Yeah, and then if we compare soil analysis with plant sap analysis, then it's interesting to see that on location 1, the phosphorus in the soil was much higher than it was on location 2. So it was almost 5 times, or more than 5 times higher on location 1. And that's what we were also seeing in the leaf analysis, that on location 1, in the Cabernet Cortis and in the Sauvignon Gris, the phosphorus uptake was over the whole season higher than on location 2. So your soil analysis can also predict or give already the first insight in how your nutrient uptake is going to be in the plant. Yeah, other points of interest on the trial location. The variety Cabernet Cortis is very sensitive for bunch stem necrosis. And if we look to the actual uptake of the magnesium on location 1, then we see, compared to our target value, so this is the minimum target value and this is the maximum target value, those gray lines, that magnesium has stayed way too low or compared to our target values over the whole season. And eventually, in the end of the season, yeah, the bunch stem necrosis was clearly visible on the leaves. So the leaves showed a magnesium deficiency, and it showed the bunch stem necrosis on the trusses. On that moment, we also took an extra plant sap sample to see if the magnesium was much different compared to the other measurements over the whole season, but it followed the same trend of low magnesium. So if your variety is sensitive for bunch stem necrosis, keep an eye on your magnesium in plant sap. So if you get below our target values, there's a big chance you're going to get problems with it. Yeah, and then the fruit analysis. First of all, if we look to total nitrogen versus pH and sugars. So we saw on location 2 in the Sauvignon Gris that the total nitrogen was much higher over there. And if we look then to the fruit analysis and we look to the measured total nitrogen, then we also see that on location 2, the Sauvignon Gris showed much higher levels of total nitrogen. So yeah, basically as expected, if you find higher levels of nitrogen or if you find higher levels of potassium, for example, in your leaves, then most probably, well, for sure, you're going to get higher levels in your fruits as well. Other thing I wanted to show, yeah, and the question is if that also led to a higher pH. So the higher total nitrogen showed in this example also a higher pH in the fruit samples and lower sugars, but the difference is quite small. But the difference in total nitrogen is clearly visible as well as we saw in the leaves. Ammonium, same story. Ammonium much higher in the Sauvignon Gris on location 2, and as well in the fruit analysis we saw much higher levels of ammonium. And that might be interesting for table grapes to keep an eye on, because higher ammonium can be associated with a negative influence on shelf life. So definitely something to keep an eye on, that your ammonium is not getting too high in the leaves, because it will also get higher in your fruits. Yeah, fruit analysis, potassium, nitrogen versus pH. Yeah, over the whole season we didn't saw a big difference on potassium, and also not a big difference on total nitrogen on the Cabernet Cortis, and also in the fruit analysis. In this case, total nitrogen was even exactly the same, and there wasn't a big difference between potassium and uptake as well in the fruits. Only the location where we had the stem necrosis, we tested that one separately. That's where we saw lower potassium and lower total nitrogen, and also the pH over there was lower. So that seems to follow the same trend as what we saw in the leaves. Fruit analysis, potassium, nitrogen versus pH. Yeah, we saw a difference on potassium and a difference on total nitrogen in the souvenir gris on location 1 and location 2. And yeah, also that is visible in the fruit analysis. So on location 1, the potassium was higher, and that's what we saw as well in the fruit analysis. But on location 1, the total nitrogen was much lower, and that's what we saw as well in the fruit analysis. Now, and if we now look to the pH, yeah, then it's interesting to see that the pH was lower there where the total nitrogen was much lower as well compared to location 2. So in this case, so it might be different on other fruit analysis or other occasions, but in this case, the total nitrogen seemed to have a bigger influence on the pH than the potassium had. But for sure, we saw it already with the carbonate cortis. The higher the potassium, normally the higher the pH in the leaves, and the same we saw then in the fruit analysis. And phosphorus. So we saw higher phosphorus on the souvenir gris and the carbonate cortis on location 1, and that was also the location where we saw in the fruit analysis the highest phosphorus levels. So that corresponded also good with what we saw in the leaves. Yeah, then our deficiency level research. Yeah, one of the most seen deficiencies in grapes is a magnesium deficiency. So these are 6 different symptoms, but all are magnesium deficiency. And what we basically did is that if we saw deficiency symptoms, we took still healthy leaves, we took leaves where the symptoms were already clearly visible, and we took leaves with very clearly visible magnesium deficiency, like in this case. We took plant sub-analysis of it, and we compared the values with each other, because that helps us to figuring out, okay, what's the real minimum level on where you can expect to see magnesium deficiency when you get below that level. Here's another example from healthy to deficiency symptoms, and yeah, also there you see that the lower the magnesium goes, yeah, the more clearly visible the magnesium deficiency. So yeah, if we take those 6 leaves which we have sampled, then it seems to be that somewhere if we get below 450 or 480 ppm, that you can expect a magnesium deficiency. So yeah, that's important. Basically, it's good to keep an eye on that you keep your magnesium high enough and good within the target values, because if you get below the target values, there's a big chance you will start seeing the first symptoms of magnesium deficiency. We also look to iron deficiency, because that's very common to see in grapes. And we saw on these examples in the young leaves, because this one is the young leaf, this is the old leaf, that if your level is getting below 2, that seems to be the level where to expect the first iron deficiency symptoms visible. But more study is necessary to figure out, yeah, on what level iron levels will be okay and that it's not deficient anymore. Manganese deficiency, because sometimes, or basically manganese can look quite similar to magnesium deficiency, but by doing a plant sub test, we saw that magnesium levels were still good within the leaves, but that the manganese were or was too low. So this is showing from healthy to visible manganese deficiency. Another manganese deficiency symptom clearly visible. So you see the nerves are still green, but around it, it all turns yellow. And this is a combination of manganese and magnesium deficiency. So both nutrients were too low in uptake in the plant, and that was also visible on the leaves. So this is a combination of magnesium and manganese deficiency. So that was basically all the research we have done in this season on grapes. I hope you found it interesting. Keep following us on YouTube, LinkedIn, or Twitter. And if we have more information or results to share, we will do that on our social media platforms. If you think, okay, I'm interested more in the relation in plant nutrition uptake, yeah, we give courses like a webinar where we explain the antagonistic interactions between the nutrients. We explain more about the mobility of nutrients. There's a part about plant health and factors that have influence on the nutrient uptake, and how to interpret a plant sap report. So check our website for webinar options. Now, if you want to have a webinar, and that's mostly 2 sessions for yourself, and that can be grape specific, or a seminar on location, that's also possible. So you can always contact us for that. So yeah, again, hopefully this was interesting for you. Take something to learn from it, and yeah, we might keep in contact. Thank you for looking to this video. Thank you. Bye.